Hi, welcome to Smart Solver. Let's learn how to study smart. Understand deeply, practice daily. Then you'll solve confidently. You can succeed anywhere.
Welcome to this explainer. Today, we are literally zooming in, way in, to explore the hidden universe right under our noses. We're talking about the fundamental building blocks of life itself. Let's get right into it. Real quick, take a second and just look around the room you're in right now. Look at your desk, the bookshelf, or hey, maybe there's a sunbeam coming through the window. Do you see any dust? It's pretty much everywhere, right? Well, here's a wonderfully gross fact for you. Most of that household dust floating around, it's actually dead cells. Yeah, that dust largely comes from living things inside your house, just shedding tiny pieces of themselves all day long. Kind of wild, right? But to actually see these individual pieces clearly, our human eyes just aren't strong enough. We're going to need some help.
Part 1. The hidden world around you.
So, let's establish exactly what we're looking at. The crucial distinction here is between a cell and an organism. All living organisms are made of cells, which are the absolute smallest units of life. What's truly fascinating here is the scale of it all. Some organisms, like bacteria, consist of literally just a single cell. But humans? We are massive, formed from trillions of cells all working together. Just to help wrap your head around exactly how small these microscopic building blocks are, think about the number 100. If you take a pencil and draw a tiny period on a piece of paper, you could fit about 100 animal cells straight across the width of that dot. I mean, 100! It really puts their microscopic size into a completely different perspective. Moving on to part 2, Hooke's tiny cork rooms. Okay, so how did we even find out about these things? Well, check out this 350-year-old drawing. It was made by a scientist named Robert Hooke. About 3.5 centuries ago, Hooke looked down a very early microscope at a thin slice of cork, which is essentially just tree bark. And what he saw were these tiny, repeating room-like structures. Because they reminded him of little empty rooms, he decided to call them cells. Those right there were plant cells, and that one simple observation completely changed biology forever. Part 3. Your superpowered eye. To unlock this universe, you need a superpowered eye, and that tool is the microscope. It's an instrument specifically designed to let us see very small objects by magnifying their image using special lenses. When we use this tool to look carefully and in detail at an object, we call that making a scientific observation. But remember, for the microscope to actually work, the object usually needs to be super thin so light can travel straight through it. Sometimes, we even have to add color, a dye or a stain, just to make those nearly invisible structures stand out so we can actually observe them. Let's break down the anatomy of a standard light microscope. Right at the very top is the eyepiece. That's the part you look through. Below that are the objective lenses, which, do the heavy lifting of actually magnifying the object. Then there's the flat platform in the middle called the stage. That's where you securely place your slide. Below the stage, you've got your light source. And over on the side, you have what I like to call the steering wheels, the coarse focus knob for big adjustments, and the fine focus knob to dial it in and make the image crystal clear. Up next, part 4, step-by-step scientist training. All right, let's get you trained up. Following these golden rules ensures safe and effective observation. First, always start by moving the stage down to its absolute lowest position before you carefully place your object. Crucially, you want to always select the objective lens with the lowest magnification first. This gives you the widest field of view. From there, look through the eyepiece, slowly turn that coarse focus knob until the object appears, and then dial it in with the fine focus knob. Once you've got it perfectly in focus, then you can safely switch to higher magnifications to see the object in much greater detail. But hold on, a scientist's job is never done just by looking. We have to actually record what we see. When you're recording your microscope observations, always note down the magnification you used so other people can replicate your work. Draw your diagrams using a really sharp pencil for crisp, accurate lines, and definitely use a ruler to draw straight lines to your labels. It's all about precision here. Which brings us to part 5, the magnification multiplier. Figuring out how much you're actually magnifying an object is beautifully simple. To find the total magnification, you literally just multiply the eyepiece lens by the objective lens. So for example, if your eyepiece magnifies by 10 times and your objective lens magnifies by 20 times, well,
10 times 20 gives you a total magnification of 200, meaning you are seeing the object 200 times larger than real life. It's really that easy.
Okay, let's put that into practice. Imagine you're in the lab observing an onion cell. The eyepiece lens has a times 10 magnification, and the objective lens you're using is set to times 50. Quick math, what is your total magnification?
Yep, you got it, 500. You would be observing that tiny little onion cell at 500 times its normal size. That right there is the sheer incredible power of the microscope in action.
Finally, part 6, the quick revision boss battle.
All right, let's see what you've learned. First question, think way back to the very beginning of this explainer. What are all living organisms fundamentally made of, and what specific scientific tool do you actually need to observe them in detail?
The answer, of course, is cells. They are the fundamental building blocks of life, making up everything from single-celled bacteria to, well, you and me. And because you can fit 100 of them across a tiny full stop, the absolute only way we can observe them is by using our essential tool, the microscope.
Let's try another one. Can you remember the anatomy of that microscope we just looked at? Try to describe the specific jobs of the lenses, the stage, and the focusing knobs.
Let's do a quick recap. The lenses, which is both the eyepiece and the objective lenses working together, magnify the object. The stage is that secure, flat platform where you carefully place the slide. And those focusing knobs, the coarse and the fine, you adjust those to bring your blurry object into sharp, crystal clear focus.
And just like that, you now have a really solid grasp on how to safely handle a microscope, calculate its magnification, and reveal the hidden building blocks of life. So I'll leave you with this final thought. The next time you look at a blade of grass or even that speck of dust floating in the sunbeam, just remember the incredibly complex, hidden architecture inside. What microscopic mystery are you going to choose to observe first? Thanks for hanging out with me on this explainer.
Thanks for watching Smart Solver. Keep learning, keep growing, and keep solving smarter every day.